Hello and welcome to In Control, the first podcast on control theory. Here we discuss the science of feedback, decision-making, artificial intelligence, and much more. I'm your host, Alberto Padoan, live from a recording studio in Zurich. Quick thanks to our sponsor, the National Center of Competence in Research of Dependable Ubiquitous Automation and the International Federation of Automatic Control. Our guest today is Mustafa Khammash, Professor of Control Theory and Systems Biology in the Department of Biosystem Science and Engineering at ETH Zürich. Welcome to the show, Mustafa.
Glad to be here. Thanks for having me at your podcast, Alberto.
Mustafa, let's jump right into it. What is cybergenetics?
Cybergenetics is actually a portmanteau of two words, cybernetics and genetics. And it's a term I came up with a few years ago because I got tired of saying all the time research at the interface of systems and control theory and genetic engineering. So it just seems much more fun and much more compact to just say cybergenetics. So essentially, it is just that. It's a fusion of cybernetics, which is the term coined by Norbert Wiener in 1948, which aims to study control and communication in the human and in the machine under the same umbrella. And it has a more modern feature, which is the genetics part, because you see, at the time of Norbert Wiener, 1948, the structure of DNA hasn't even been discovered yet. And so much of the work that went on in cybernetics had more to do with things like neuroscience, maybe diseases that had to do with neuromuscular sources. But nowadays, we have much more powerful tools. We have much more powerful genetic tools, and we could do so much more. And so you can imagine now we can bring this cybernetics to a new level. So that's what I mean when I say cybergenetics.
Yeah, that's a fantastic definition. I might ask, what is a gene, actually? What do we mean by that? And what do we mean by genetics? What are the goals there?
I think a gene is essentially the basic hereditary unit. And, very simply, you can think of a gene as corresponding to a single protein. It's not exactly like that. It's not always like that, but I think this is good enough. And so each gene is a stretch of DNA encoded using nucleotides on DNA. And when that stretch is expressed, the outcome is a single protein. This protein goes on to interact with other proteins or maybe with other genes to implement various different functions.
I guess there it would be useful to have a small primer on how biology works and how DNA is used in biology or in biological systems.
Yeah, so of course DNA is central to life. In its sequences are encoded, all the biological functions that the organism requires. And it's extremely dense with information. And so you can think about DNA as encoding all the information needed for the biological processes required for life. But it's not only a storage device, because DNA itself is a molecule and it interacts with other proteins that through which this interaction regulates the amount of gene expression. So gene expression is a term that comes up repeatedly, and you mentioned something about it. So it consists of two different processes. The first one is transcription, and the second one is translation. So transcription is the process by which the DNA is read inside the cell, and the outcome of this transcription process is messenger RNA. So you have a messenger RNA that then goes on to be translated by cellular machines called ribosomes. And the outcome of that process, of that translation process, is these proteins. Usually you have a sequence of amino acids that then folds into a secondary structure, a tertiary structure. And once it's fully folded, then it's called a protein. And that's when it's functional. And by functional, I mean, it can interact with other proteins, with other RNAs, with other genes to do all the wonderful things that the cell has to do to exist.
I guess a Maybe at this point already, someone in the audience will think, am I listening to the right podcast? But you mentioned so many words that come from engineering. So you mentioned regulation, you mentioned machines, you literally called ribosomes machines, and you mentioned interaction, which is so key in our field. So what is the role of control theory here, and what does it have to do with biology?
Actually, control is very native to biology. In fact, biological systems discovered control billions of years before we did, and they have been employing it in different flavors, different levels of organization, much longer than we have. And so it's not really an exaggeration to say that much of the complexity of biological systems have to do with the requirements of regulation. And that's because regulation, or what biologists call regulation, what we also call control, exists at every level of organization in biology, from the molecular level to the cellular level, to the tissue level, to the organism level, even to the ecological level. And so if you look at the typical biological systems, you will very quickly find tons and tons of feedback loops, feed-forward loops that are helping the cells achieve their various functions in a very robust fashion. So really, it's quite natural to be doing control in biology because, as I said, much of the complexity of biology is about regulation or comes from this regulation.
Yeah, and I was actually looking at your biography, in doing my homework for this podcast, and you're natively a control theorist, but you then ventured, if you want, in this field. And so I was wondering what has drawn your attention from being a very theoretical person who was working into deep questions in robust control towards this fascinating field.
Yeah, that's a very good question. Actually, thinking back at those times, this was in the late 90s. So I was working on robust control for my PhD thesis, and the early and mid into the late 90s, I was still working on robust control. But by that time, really the main problems in robust control have been solved. So the H-infinity problems, the state space solutions, Turicati equation problem has been solved. And there wasn't much left in terms of, big problems. And at the same time, around the same time, my wife had our second child, and after childbirth, she had the problems with her thyroid gland. So she was going through a period of hyperthyroidism and hypothyroidism. So the thyroid hormones were essentially oscillating like crazy. And I had no idea what was going on. So I went to the library and I picked up a textbook on endocrinology. Endocrinology is the science of hormones and their actions. And really, this was a very transformative moment because I was shocked by what I was reading. Everything in that book seemed like it's a control theory without the equations, right? So it's one exquisite feedback loop after the other, doing all kinds of different things, but of course, no equation. So I was really fascinated by this, and that led me to go to the National Animal Disease Center at Iowa State University, where I was at the time in the US. And I talked to one of the physiologists, and I said, I'm a control theorist. Please, can you give me a problem where you think control theory could be useful? And it was very naive, but actually it turned out he gave me a fantastic problem. He gave me a problem that he was working on that had to do with how mammals, in his particular case, dairy cows, regulate their blood calcium. Okay, so us humans and other mammals, we have a very tight regulation of calcium in the blood plasma. And this is important because calcium ions are needed for muscle function, nerve function, blood clotting, signal communications, and so on. So it's a very important biochemical role. And because of that, we have evolved a very elaborate control system to maintain the concentration at about 9 milligrams per deciliter. So it's really a fascinating problem, and it couldn't have been a better problem to start with. In that case, they were looking at a specific disease that dairy cows had that humans don't usually have. It's called milk fever. And it turned out to be sort of some kind of a dynamic instability in the closed loop system that was induced by the way humans breed cows to make more and more and more milk. And so myself and another young master's students that I had, we had a lot of fun working out the details. And when all the dust settled, we had discovered that mammals regulate their blood calcium by using proportional integral feedback. It's as simple as that. So that was kind of a nice discovery. All the ingredients and the elements were there in terms of the main players, the hormones and their interactions. It just waited for somebody with a control perspective to explain what these interactions were actually doing.
Well, you probably didn't have any mathematical model for studying this phenomenon, or was there actually any? Did you have to develop everything from scratch?
We didn't have any mathematical model, but we developed a very, very simple model, actually. Just looking at the blood volume as a constant volume and looking at various sources for calcium and various sinks, and then making simple arguments like, disturbance looks like an outflux of calcium, and then we know the system regulates with zero steady state tracking error, so the controller must have such and such properties. And so the model we used actually was very simple, maybe a second order model. Only when we were looking at the specific disease did we have to go to more complicated nonlinearities. And even then, there were saturation type nonlinearities, which were very naturally justified based on the nature of the disease that we were working with. So basically, third-order linear systems with saturation nonlinearities. That was sufficient to explain the whole story. Of course, one can develop more complicated models with different compartments and so on. But I think for the purposes that we needed it, this was sufficient.
But the essence was there. Yeah. And so that was your, let's say, initial start in this world of systems and synthetic biology. Actually, may I ask, what is the difference between the two fields, between synthetic biology and systems biology?
That's a very good question. I get asked this question a lot. I think, one analogy would be it's similar to the difference between system theory and control theory. Yeah. So systems biology aims to analyze existing biological networks, and it's some kind of a reverse engineering, if you will. So you have a network that's functional. You have a cell that's doing a certain thing. You're trying to understand why it does what it does, and when it fails, why it fails. And so you kind of ask questions about a network that already exists. Now, synthetic biology does things a little bit differently. So it's more like a forward engineering. How can we modify this network? How can we add additional components so that it either achieves new function or it fixes the function that somehow was broken, right? So if there's a disease and then you add a new element into your cells to kind of cure that disease, this would be more on the forward engineering part. So I think that's really the main difference. It's like adding the synthesis aspect to systems biology.
In our language, the former would be analysis, dealing with analysis, and the second would be dealing with design. So they go hand in hand. They're complementary.
That's a very good way of saying it. Also, synthetic biology can be used for analysis. So, for example, let's say you have a hypothesis about a biological system. You think it works in a certain way, and then you go build it in the lab. And if it doesn't function the way it's supposed to function, then you have to adjust your understanding. And so one can also think of synthetic biology as sort of a field that allows you to tinker and get a deeper understanding of the underlying science, not only just to build new things.
And this is reminiscent of another Feynman quote, building is understanding, that appeared on this podcast before.
Yeah. What I cannot build, I do not understand.
Exactly.
Yeah,
exactly.
Yeah, exactly. Yeah. That's an often quoted quote by Richard Feynman, by synthetic biologists.
Yeah.
Yeah, and I think it captures the essence quite well.
Actually, I was looking at the history of synthetic biology, and it seems to be extremely recent, at least, the year 2000 seems to be the turning point in this field. Of course, there are predecessors, but in 2000 there were, I believe, 3 really milestone papers on Nature where people actually implemented biological circuits that essentially implemented a switch. Another one would implement an oscillator, and another circuit would implement a feedback mechanism, essentially. So all three in the same year. It's incredible.
That's right, even in the same journal. Exactly right. So there were 3 milestone papers that essentially demonstrated what you said. One can think of these circuits in the following way. One of them used 1 gene, and the second one used 2 genes, and a third one used 3 genes. And they all had negative feedback. So when you have negative feedback with 1 gene, you have an autoregulatory circuit. So that's the usual feedback systems that we like. When you have 2 genes, each having negative feedback on each other, so you have positive feedback, and that's what implements bistability, in this case, a toggle switch. And the third had 3 genes, each repressing the other in a sort of a ring type fashion. So then you get a ring oscillator. And yeah, that's work of Michael Elowitz. So yeah, those are the 3 classic papers that kicked the field into motion. And in fact, the tools of synthetic biology, genetic engineering, the most important tool, they were already available in the 70s and 80s, and many biologists actually have done synthetic constructs. But I think in the year 2000, people were thinking of this field more like VLSI circuit design, right? So you can build circuits and devices that then you can put together into functional units. And this is a distinctly engineering approach. It's no longer just tinkering with 1 gene or another to see what happens, but it's actually with a purpose to build more complex systems. And here we are, 23 years later, and we're still struggling with a lot of these designs. We can build more complicated circuits now. Instead of 1 or 2 genes, we can build circuits with tens of genes. But there's still lots of challenges. So I think the field is still in its infancy. A huge promise, of course, but lots of challenges. So I think for researchers who want to work in this field, I think this is a very good time to be doing it.
There's a lot to unpack in what you just mentioned. I don't know where to start. So one of the things that make me think when you were elaborating on your thoughts was that you touched on so many elementary components that are very familiar to the area of computing. So switches, oscillators. So in hearing about these concepts, what I think about is a computer. So it seems like it's possible to assemble some sort of biological computer out of those components. The second thing that I'm extremely excited about is what we can actually do with these circuits, so the ones that you just mentioned, and what is actually the time horizon in which we can hope to see something, let's say in our lifetime. Is it going to be in 2 centuries, a millennia? Who knows? And the third thing that I would like to ask you is, what are the main developments in the middle, let's say from 2000 to today? So what happened in between?
Okay, so starting from the last part of your question, so what happened in between? I think that what happened is people started to build more complex circuits, and in the process, they ran into challenges. So there are various challenges when you're working with biological substrates that one does not find when working with electric circuits. Interestingly, a lot of people working in this field, synthetic and systems, synthetic biology in particular, have a background in physics or engineering and so on. So the electric circuit analogy has been a running theme throughout. And I think while this analogy had served the field well early on, I don't personally think it's any longer a useful analogy to continue to pursue for reasons we can discuss if you want. But the idea there was, you want to modularize everything. So you want to build functional units that then you can combine together with other units in series or in parallel and be able to scale up that way. But biology didn't play well with this vision. There are many issues. For example, when you're doing electric circuits, communication or information is transmitted through conduits, wires, that are very, very precise, and only the parts that talk to each other generally talk to each other, right? I mean, of course, you could have electromagnetic interference and so on, but most circuits don't have these issues. In biology, it's not like this. All the molecules see all the other molecules. So the way you would transmit information is through specificity. Molecule A binds to molecule B. Maybe it binds molecule C a little bit, but really it's molecule B that it needs to interact with. So you end up with a lot of crosstalk like this. So that's one problem. Another problem is noise or stochasticity. So when you're working with electric circuits, you have your Ohm's law, you have your ordinary differential equations, or everything is more or less deterministic. Of course, one can still talk about shot noise and so on at various frequencies, but in biology, it's not like this at all. So molecules very often come in very low copy numbers. So you have one gene, for example, a handful of mRNAs, and these interactions between these low copy numbers species, they're random in nature. Molecule A interacts with molecule B only if they happen to collide with each other. But maybe in the process or before that happens, molecule A interacts with molecule C first, and then some other reaction takes place. So really, the timing and order of chemical reactions when you have very low copy numbers is random. So this should be compared with what you've learned in chemistry in high school, where things are present in very large copy numbers, Avogadro's numbers in a test tube, where none of this is true. So you can actually approximate, or you can safely describe the dynamics using ordinary differential equations. They're nonlinear, but that come from mass action kinetics or other type of kinetics. But in the end of the day, you get deterministic equations that represent continuous variables that describe the different concentrations of the different reactions. But not so inside a living cell, where the copy numbers is low and the randomness is ever present. So a more suitable tool in this case is to look at, instead of continuous variables that evolve deterministically, you look at stochastic or random variables to represent the number of molecules for each of the different proteins or RNAs, and the stochastic nature in which these random variables evolve. So really, the typical model that we work with in this case is a continuous time discrete state Markov process, and that's a much better representation of what's going on inside a living cell, especially when you have very low copy numbers. So noise is another big problem that one has to deal with. Of course, nonlinearity as well, because everything is nonlinear when you have chemical reactions. And so a wealth of mathematical as well as computational problems to deal with.
Yeah, so in what you just said, essentially you replied to a question that I wanted to ask you, which would be, what are the challenges in controlling biomolecular systems? And to me, it seems like the scale, in a way, determines also the way you describe the processes that you're interested. As you said, there seems to be a fundamental difference between electrical circuits and biomolecular, if you want, systems. So, and I wonder, what is the role of control engineering there? So what can we hope to achieve? What is the current progress there? Mm-hmm. And where are we going towards?
Mm-hmm. I think I don't want to give the impression that one must only use these stochastic models, otherwise nothing works. Because depending on the problem that you're looking at and the scale that you're looking at, very often deterministic ordinary differential equations can still serve as good models for biological phenomena. Just one has to be careful that when you're looking inside the cell, especially smaller cells like bacteria, this assumption may be violated, and you may end up with behaviors that are very different. Very often, actually, the most suitable approach is a hybrid one, where you have some variables are stochastic and discrete, others are continuous and evolve deterministically, and they interact with each other. So, this is probably the most useful model. So what control problems exist? If one sticks to the deterministic framework, and let's talk about this first, the issues that I mentioned before, crosstalk is one of them. You would like to do regulation. You'd like to regulate certain variables, and these variables, for example, must track certain set points or reject certain disturbances. So you end up with a control problem. How do you design a feedback or feed-forward mechanism that will implement this, or that would meet these control specifications? So what is so challenging about biology? Well, for one thing, again, we're sticking even to deterministic descriptions. All the variables are positive variables. So whatever you want to do, it has to be done with positive quantities, right? So positive systems. I mean, that's one of the main challenges. The functions that you build, the controllers that you have to synthesize, you cannot just write a transfer function and just say, please build me this transfer function, or this nonlinear function. You are constrained by chemical reactions. So everything that you want to implement, your controller, for example, has to be implementable using biochemical reactions, right? So that also adds a second constraint. And it's the interplay of these two constraints, the positivity of the variables plus the constraints that are imposed by the type of interactions you can have inside a living cell, I think give biology the distinctive flavor that it has. And it opens up a wealth of problems in control theory. What kind of controllers can you build using these chemical reactions? How would you achieve, let's say, perfect tracking, robust steady-state tracking with these type of controllers and these type of networks? They have to be positive. They have to be implementable, realizable as a chemical reaction. Can you realize any transfer function, or can you approximate it using biochemical reactions? So there's really a wealth of problems that control theory can contribute to. And I'm not saying that existing control theory can address these things. And that's really the beauty of this, is that it's not just, here, I'm just going to move the methods that people have already developed in our field and just apply them to control theory, to biology. That would be the wrong approach. I think one has to be open-minded, and you look at the problems that emerge in biology, and more often than not, you have to develop a new control theory that is suited for biology. And I think that's even more fun that way. So that's what we do in my lab. We try to develop a control theory, a new foundation for control theory at the molecular scale. And then we also try to build these chemical reaction mechanisms using genetic engineering tools. And so I think for me, at least, the challenging part is also the most fun and interesting part, is where the theory and the experiments meet. The theoretical questions are motivated by the experimental considerations, and the experiments we do are always guided or informed by the theory we develop. And there's this interaction, very close interaction, that I think is just, every time theory impacts the experiment, I really get a kick out of this in the lab. And I think that's sitting at the boundary between the experimental world and the control theoretic world is really a very good place to be, at least for me.
Sounds incredibly interesting. I just want to comment on what you mentioned before, so about the constraints, if you want, that biology imposes. So maybe just to clarify the fact that all variables need to be positive, I guess that's because we're always talking about concepts that are quantified quantities of things that are out there, or I guess fluxes or other concentrations.
Yeah.
Exactly. So and it's incredible how positive systems theory there has a huge role. And the second thing that I wanted to mention is related to the question of realization that you mentioned before. I find it very fascinating that in this context, you're very much constrained, as you mentioned, by chemistry. And so the realization theory, if you want, that you can build out of these systems has to comply with those rules. And I guess it imposes huge constraints because everything has to comply with the law of mass action or, with those nonlinear equations. And therefore, it's very, very different from, again, the realm of electricity. But I also thought that maybe this is actually a good point, as you mentioned about the work of your lab, to shift gears and to start talking about motifs. And one of the motifs that your lab has developed, which I find incredibly interesting, as a preview to the listeners, we're going to talk about one of the first PI controllers, if not the first, I'm not sure about it, but I think it's the first, in biomolecular systems. And this is something that gets me incredibly excited about, the future of this branch of control theory and systems and synthetic biology.
Yeah, so Biologists like to talk about motifs. This is not a word that exists, I guess, or is commonly used in our field, in control theory. So let me just explain what a motif is. So a motif is usually just a pattern that recurs with a frequency that is much higher than one would expect if it were by chance. Okay, so you could have a motif in terms of a sequence of nucleotides in a DNA that repeats. What we're more interested in is network motifs. And so these are simple network elements that implement a certain function that you usually see much more frequently than chance, such that this small network forms a basis for much more complex biological behavior. So one example of that is a feedback loop. So that would be a simple network motif, or a feed-forward loop, or a, let's say, an oscillator, or a positive feedback loop. So those are the different motifs. When I first joined ETH in 2011, before actually joining, I was just doing purely systems biology, so more analysis, system theory. Now coming to ETH, I had the opportunity to start my own lab. I have to say, before that, I hadn't touched a test tube in my life, so it was a bit of a experience to kind of try to learn all of these techniques and build a wet lab from scratch. But I already had tenure, I had nothing to lose. And so, if it didn't work, I would just close it.
That's the way you do it.
continue doing theory as usual. And that was one of the best decisions I had made, actually, professionally, is to start a wet lab. Initially, I thought I would just have one bench with one student, maybe doing some experiments to verify the theory. Right now, I have more than 16 different benches, and my group is actually more than half people doing experiments. So it's been a really very rewarding experience. We didn't do it at the expense of theory. We just it just added to the theoretical work and gave it much more color and life and meaning. And also to the theoretical questions that we ask are now very different because they're driven by what we see in the lab. So having the ability to do experiments meant to me that now I can switch from doing system theory to doing control theory. So now we could do synthesis. It didn't really make much sense to do synthesis if you're just analyzing existing systems. Now, suppose you would like to design a new feature, or you would like to control a living cell in one way or another. We started by using a technique that had been developed earlier called optogenetics, namely developing cells that are actually sensitive to light so that you can actually control them by shining light with different intensities. That was the earlier problem. So we would still use computer controllers, but the computer controllers would send out signals that are light signals whose intensities actually serve as the actuation. That was fun, and we actually were able to build the first feedback control system, computer-controlled gene expression. At the time, this was 2011. But then another experience that I had in the past, namely integral feedback, came to the front, and the question was, can we build integral feedback controllers inside living cells? Having shown that these systems naturally exist, for example, in calcium and many other systems, it was obvious to ask the question, how would you build one? Nobody had built integral feedback controllers. And then, we started trying to build a system that mimics the calcium homeostasis, and we spent a lot of time on it, several years, 2 or 3 years, not really very successfully. One time we thought we had it, and it was exhibiting zero steady state tracking errors, everything that an integrator should do. But to our disappointment, it was actually an artifact. We discovered it was a biological artifact, and what we are seeing was not really the action of an integral feedback controller. It was a combination of negative feedback with disturbance feed-forward that was behaving very much like what an integrator would do. And this went on for quite a while, and then I think the breakthrough came from the theory side. We had come up with a new motif that, which now we call, I called it the antithetic motif for a reason that has to do with the way the molecules interact with each other. So this antithetic motif on paper should be able to implement or realize integral feedback. And it required two molecules, and you can do negative variables by having two positive variables and look at their difference, essentially. And so this motif is a kind of a sequestration or annihilation motif, to use the term we like to use in biology. But essentially, two molecules bind with each other, and they prevent each other from being active. And one can use that behavior to realize integral feedback control systems. And so the theory was nice. And in fact, it turns out that these type of integral feedback controllers are also noise resistant. So they allow us to solve this problem with stochasticity or noise. In fact, they would exploit noise so that they would function better in the presence of noise. Whereas if the system was noise-free, one would observe oscillations. Now the presence of noise would actually lead to a dampening of this oscillation. Very counterintuitive, but one can prove this mathematically. And so then there was a sudden and abrupt switch in the lab. Okay, drop everything that you're doing with these other systems. Let's now try to implement these antithetic controllers. It still took a good 1.5 to 2 years to build them and verify that they were functional. But in fact, because we had to find the right biological molecules that had the behavior that we were looking for, and it was a very stringent constraint. These molecules have to sequester each other. Once they're bound, they could not function anymore to actuate. And so we found them in the form of what's called sigma factors that exist already in biology. Once we built it, we could verify that we actually have an integral feedback controller on our hands. And I think these were very happy times, because it really, from beginning to end, it took a good part of 7 years to build that first integral feedback controller. But it was a glorious moment when this finally worked. The paper appeared in Nature in 2019. Now, to 2023, we can build many of these, let's say one every 2 or 3 months, with much better properties, better dynamic range. It works in mammalian cells and bacterial cells. So, once we had that first integrator, now we started learning about its property and how to optimize it. We have advanced quite a lot since then.
That must have felt like a real eureka moment, almost like in Harold Black developing the negative feedback amplifier.
At least for me, it was, the culmination of a lot of attempts, failed attempts, and a lot of effort. For people in the lab, it was salvation, because they no longer had to keep doing these experiments that would take a long time. You see, when you want to do, let's say, a simple time course, and if you're building a circuit, you put a oscilloscope, and then you look at the voltage output and you see whether steady state is achieved or not. In biology, steady state happens after 10 hours. And so if you want to do a time course, then you have to do samples every so often during this time course, and you have to do all sorts of dilution processes and freezing of these samples and so on, just to get one trace, only to find out that it hasn't worked for one reason or another. So it was a very grueling experience for people in the lab. And when the new theory was developed, there was another small gift that came with this theory, and that is that we could show that in the presence of noise, this motif is the only motif that will implement integral feedback. So this is kind of a necessary condition. And the reason why this was good news for the experimentalists is just a realization that this is the right motif. We're not missing another motif that could have done this easier. If it is to work, this is the only way it would work, right? So this was completely reassuring. And it was just a matter of time when they got the circuits to actually work the way we were expecting that they would work.
This is incredibly exciting, at least for me as a follower of this story. It's fantastic. I would like to dissect so many of the things that we touched on just in the last few minutes, because we inadvertently touched on, the enabling technologies that allowed this eureka moment to happen. We also touched on the concept of antithetic integral feedback that I would like to dig into a little bit more. Yeah, maybe we can start from the former. Initially, you were speaking about optogenetics, which, pun intended, allowed you to shed new light on these biomolecular phenomena. Maybe we can talk about the enabling technologies that allowed control theory to actually implement these circuits.
Yeah, I think when I think of cybergenetics, at least the way I've thought about it, when coming up with ways to do cellular control, one can think of two different flavors, right? So general flavors. One flavor, which is using computers to control living cells. So computers in the loop. There, implementing any control system you like is easy and possible. The challenge there is to interface the computer with the cell. So what do you need for that? Well, you need sensors and you need actuators. In terms of the sensors, we would like to measure what the cell is doing. Let's say how much a gene is expressed. I mean, this problem had been worked out. There was a topic of a Nobel Prize, 2008, in chemistry. That's the well-known fluorescent protein, the discovery of the green fluorescent protein. The main idea there is very simple. If you have a protein that's being expressed by your gene, what you do is you genetically engineer your gene such that in addition to that protein, it fuses another protein that fluoresces. So when that original protein is expressed, attached to it is another fluorescent protein, and by measuring the intensity of this fluorescence, you get an idea about the abundance of the expressed protein, and so that's one way to measure. Initially, this was green fluorescent protein, but I think it wasn't long before other wavelengths were developed, and so one can look at multiple proteins simultaneously, so you can do even multivariable control if you want. But still very challenging with a single output. So the idea, though, we can measure protein abundances. So that solves the problem of sensing. For that, you could either have your cells under a microscope, and then you would image it, and you would see how much fluorescence that your cells have and quantify that. Or there's another device called a flow cytometer, which allows you to put a cell through a microfluidic channel, shine a laser on it, see how much fluorescence. So you can count the fluorescence of each individual cell, and you could do this for hundreds of thousands of cells a minute, so you can get a nice distribution for your population. You can think of this as a snapshot of the probability density function of the expression. So you can measure, you feed that to the computer, and then to actuate was a problem. So how would we tell the cells how to express, how much to express based on what the computer computes? There, I think we found optogenetics, and that was a technique that has been developed recently. At the time, it was recent. I guess now it's no longer recent. So it allows cells to be light sensitive so that the more light you shine on the cell, the more the gene is expressed. Okay, so this is an actuator. We had since developed our own optogenetic systems in bacteria, in yeast, and in mammalian cells that are particularly suited for control applications. But at the time, we just used what was already out there, and it was sufficient. Now you could do sensing, you could do actuation, then you can close the feedback loop with the computer in the loop, and then you can control gene expression, which is something we managed to do in 2011. Since then, we've been using this technology to control cells in a bioreactor so that we can optimize various protein products and various other applications. So this is one flavor where the computer is in the loop. The other flavor, which is actually in many ways is a bit harder, would be the controller is in the cell, right? So you have to genetically engineer the control system so that once you have a genetically modified cell, when the modified genes are expressed, they produce proteins that interact with each other in a way that realizes your control system. So this is what we had to do with the integral controller. We had to modify genetically the cells. In that case, it was E. coli at the time, so that they express these sigma factors that aren't normally there in that particular bacterium. It comes from another bacterium. And such that when these proteins are expressed, they implement this antithetic motif and in the process close the feedback loop. So that's the genetically engineered controls, and that's the second flavor of feedback control.
Thanks for that. You're doing an excellent job in clarifying at least to me, how should I just visualize even systems that you're acting on? So I guess in my imagination, at least, I'm visualizing a microscope endowed with some tool that allows you to shine light onto.
We have our cells in little containers that shine light through them.
Yeah.
And in that particular case, we had to develop new technology to allow us to shine light on individual cells. And that's kind of, again, using MEMS technology, microelectromechanical systems. So technology that was developed for projectors. And these are called digital micromirror devices. So these are, you can think about it as an array of millions of micromirrors that each of which can be controlled with an electrostatic field. And in this way, you can point the mirrors any direction you want. So you can shine light on a single cell or on even a subcellular compartment with the intensity that you want. So this allows us to very, very precisely target individual cells, and we can have a controller for that one cell. You cannot see it with your naked eye, but there's a controller in the computer running in feedback for that particular cell, and you can do this simultaneously and in parallel for thousands of cells. And so that's, again, brings in technology to allow us to do the feedback control of individual cells.
I was about to mention that. So there is an immense challenge also from a technological perspective in order to implement actuators, as you say, and sensors, which seems to be something easy, as we, at least you make it sound like it's something easy, but it's definitely not. It's a huge component of your work, I guess. But I also found very interesting what you mentioned about controllers that are acting in living cells. And I was wondering, what do you use as media to create those controllers? Should I imagine viruses acting into a cell, or how does it work?
So how do we introduce our controllers?
Exactly.
Yeah. I mean, there are different ways. So in the lab, what we normally use, we use electroporation. So a process by which you subject the cells to an electric field, and their membranes become more, allows you to introduce genetic material inside the cell. So this is by far the most common way, electroporation. And that works reasonably well. So it allows you to introduce new genetic material inside the cell. In a lot of the treatments that people do, if you want to access, let's say, cells in a living human or in a mouse model, there are different vehicles. So one can think of using viruses, lentiviruses, as a vehicle for delivering genetic material. And viruses have evolved, to be very efficient and effective at penetrating host cells and to deliver their own DNA to infect the cell. So now people can, or biologists can exploit that feature. And instead of having the native DNA of the virus, they introduce their own, let's say, therapeutic genetic material into that virus that then goes on to deliver the material. So these are some of the tricks by which you would introduce novel genetic material that then gets expressed into your favorite circuit, synthetic circuit.
That's incredible. Mm-hmm. Again, it sounds like science fiction, but it's incredible that we're actually able to do such things. I'd like to take a step back before we move on towards something that I find incredibly exciting. So the applications of all these ideas, what can we hope to achieve with these tools? Spending the last few words maybe on the antithetic integral feedback motif. First of all, I'd like to understand the reason of calling it antithetic integral feedback. And then I'd like to understand how it works a little bit from a control theoretic perspective.
Mm-hmm. Okay. Without equations. Oh, okay. Okay.
I understand it's a huge challenge, but we can try.
Yeah. Okay. So why it's called antithetic? Well, that goes back to the way this works. So at the basis of this antithetic controller are two controller molecules. We call them Z1 and Z2. One molecule, Z1, gets expressed at a constant rate. Okay. And it's stable, so it doesn't degrade. So you're just producing this molecule at a constant rate. The other molecule, Z2, acts like a sensor. And so the rate of production depends on the abundance of the molecule you want to regulate. Okay? So the rate of synthesis of Z2 is proportional to the variable, let's say X, that you want to keep constant. And now the reaction, the key reaction underlying this motif is a reaction in which one molecule of Z1 binds to another molecule of Z2, and they mutually annihilate each other. Strictly speaking, they don't have to annihilate each other. They could still be bound as long as the complex is not functional. Things will still work. But let's just say they annihilate each other, okay? 1 to 1. So this process is, so the two molecules are in some ways antagonistic to each other. Thus the name, antithetic control. In fact, when I called it that, I didn't have any idea that actually many of the motifs we will find out later on to implement this are things like sigma factor anti-sigma. They were already called like this, sigma anti-sigma, toxin anti-toxin, holon anti-holon. These are all pairs of molecules that have exactly the property we want for realizing this motif. And so it was appropriate to use that antithetic terminology. It was fortunate that we used it at the time. So how does this work? So if you think about this operation, Z1 and Z2, and they have this property that they bind with each other. If you have an abundance of Z1 and an abundance of Z2, and let's say they bind together 1 to 1, then what's left is the difference, right, between Z1 or Z2, if it's positive or... So this allows you to do this positive-negative type of thing. And the integration happens because of the nature of the operation. These things are being produced at a dynamic rate. And so when you subtract the dynamics of production of Z1 and Z2, what you're left with is the difference between them. The dynamics of the difference is what actually implements the integral of the error. So the difference of two positive quantities can be positive or negative. So this allows us to also overcome this issue with the sign. That's roughly speaking.
Okay. And
I can show you in the in 30 seconds if I write the equation. But yeah.
No, of course, there will be a link in the description to everything that we talked about. So, of course, the Nature paper from 2019, but also a wonderful, I would call it an overview or almost a manifesto paper about cybergenetics that appeared on the proceedings of the IEEE in 2022. I just wanted to clarify for terminology for the audience of control theory. What does it mean actually to annihilate and what does it mean to bind? I know that these are very naive questions, but it might be worth just mentioning what those mean.
Yeah, I guess I should have explained those things because I myself had struggled with these concepts when I was first learning. So binding is the process in which two molecules combine and form a complex. So A plus B gives you C. C consists of both A and B. So this is the binding event. And now the bound species may have different properties, right? It could be inactive, whereas A and B are on their own active. When they bind together, they could annihilate. So they could inactivate each other. Annihilation just simply means that they mutually degrade each other, right? So that the protein is no longer functional. Maybe ideally it gets broken down into amino acids that get recycled. Very often that's not what actually happens. You don't really need it to be annihilated. It's just an idealization. All they need to do is just deactivate. They just need to be non-functional in the complex form.
Should I picture it as the concentration exponentially decreases of this substance, or what do you mean by it's no longer functional?
Like, let's say, for example, Z1. Z1, in order to close the feedback loop, has to activate another gene that closes the feedback loop. Now, when it's bound to Z2, it can no longer do that. So it no longer acts as an activator. It's inert, if you wish, biologically inert. Whereas before the binding event, Z1 is a functional molecule that's a variable that activates other variables and closes the feedback loop. Now, once it's in that complex form, it's out of the picture. You could just completely ignore it in terms of the chemical reactions. It's no longer functional.
So while we're on this subject, I also want to touch on two other things that I believe are incredibly exciting. So one of them is that, of course, after 2019, you moved forward and you added derivative action as well. Proportional and derivative action. So at the moment, we do have a real PID controller for biomolecular systems. Am I right?
Not quite. So we have since built PI controllers and demonstrated them in mammalian cells, which is always important because the way we want to use them for medical applications. We've also come up with all the right motifs that one would need to implement the derivative aspect of part of the controller. But we haven't, to date, demonstrated a PID. We have demonstrated a PI, but we're working on the last bit, the D. Admittedly, it's less important than the PI does most of the work that you need. So it's just, I think, it would be nice and satisfying to have the derivative component. This is another question. How would you implement different derivatives using biological component? I mean, it turns out if you know how to build an integrator and you put an integrator in a feedback loop with a unity feed-forward path, then, so if you have an identity for the forward loop and then an integrator in the feedback loop, what you end up with in the transfer function between the input and the output is actually a differentiator with a low-pass filter, which is exactly what we need. We need the low-pass filter as well. As you know, all differentiators have low-pass filters in practice. And so having implemented an integrator, we have the ingredient that's needed to implement a differentiator. It's not the only way, but it's actually one way to do it. And it's just, I think, a matter of time when this finally works. I think for applications, as I mentioned, it may not be necessary. There are reasons why you want to have a differentiator, similar to the reasons why control engineers in the field use PID and not just PI. You can get faster response and good transients and so on. It could also reduce variability, so cell-to-cell variability. But strictly speaking, it's not necessary. In fact, working in the field with a company when I was back in the US, I noticed that very often the engineers have PID controllers, but they actually have turned off the D part. They just use it as a PI.
I guess 95% of the real PIDs actually do work like that.
Yeah.
I find very, very interesting the fact that the conceptual analogies that you do make with transfer functions actually work in practice. So one is guided by those concepts to realize something that is physically very, very different to the ideas that we have in mind. I also thought that before, moving towards the applications and the future of this field, it would be interesting to talk about the actual systems that you do work on. You mentioned that mammalian cells are something that is important for demonstrative purposes, but generally that's not the only type of cell that you do work on, right?
Yeah, that's correct. I mean, normally in this business, there are a few organisms that serve as model organisms where, the genetics have been worked out. There's a lot of tools that people have used. And so, people tend to work with those model organisms or model cells because life is much easier that way. I mean, biology is already hard enough. Why make it even more complicated by working on some exotic bacterium that, perhaps the genetics of which haven't been worked out yet? And so some of these model organisms are things like E. coli, right? So E. coli is probably the best understood living organism, more than us. Yeast, budding yeast, so the baker's yeast, is another model organism. That's kind of has a nucleus, unlike the bacteria that doesn't have a nucleus. So it's called the eukaryotic cell because of that. And of course, us, that's mammalian cells, right? So the sizes of these cells differs drastically. So with the E. coli being the tiniest, and then the yeast in between, and the mammalian cells are huge in comparison. But also the genetic challenges are also different. So mammalian cells are more complicated to work with. They take more time. Reagents are much more expensive, but of course they're much more important, right, for us. So if you're thinking about therapeutics, even if you want to do things like antibody production in a bioreactor, you still need to work with CHO cells, and those are mammalian cells. So the evolution of my lab started from E. coli. It was the simplest organism. It was the easiest to do genetic tests. It was the fastest and the cheapest. And then as we gained more confidence, we felt that if we are to have an impact on biomedical applications, we had better tackle the mammalian cells. And so gradually we added yeast cells and as well as mammalian cells. And now I think most people in my group work on mammalian synthetic biology, with all the challenges and also the opportunities that come with that.
So, and what type of mammalian cells, if I may ask?
So there are different types we work with. We work on cells called HeLa cells. These are one type of cells. Another type we work on are called HEK cells. And then we also work on stem cells, so mouse embryonic stem cells. So those are the three cell types that we mostly work with. And I think we will start soon working on CHO cells. So these are hamster cells, hamster egg cells.
Okay. Yeah, so maybe now it's a good moment to shift gears and move towards the future. So the applications of this field that you dubbed cybergenetics, what can we actually achieve? I mean, I know that there are things nowadays that are science fiction almost. We were chatting yesterday just before recording this episode that we can nowadays control the firing, individual firing in nerve cells of mice or rats, I forget. So the ability to engineer living cells opens up quite a bit of possibilities.
Yes, that's right. I mean, one can think of it in different ways. I mean, you can think of agriculture. Of course, genetic engineering methods are well known in agriculture, but also industrial biotechnology. So this is one area that I'm also excited about. You know, you would like to be able to make a new protein, maybe more effectively or cheaply, and you would like to do it in such a way that you can control the different enzymes and the rate of flux at different pathways so that you can get just the optimal amount of protein expression. Now with cybergenetic techniques using light, we can do this. We have recently demonstrated that we can produce more, have a higher yields by simply closing the feedback loop. And in this case, one can measure, for example, the amount of stress a cell has through this fluorescent protein technology that I told you about, and use that information to decide how much expression a particular gene should have, right? So if you have a gene that produces a product of interest, a protein of interest, overexpressing it is not the best way to get more yield, because then the cells get stressed and actually your yield goes down. And if you underexpress, again, you're not exploiting the maximum capacity of your cell to produce that protein. There's a sweet spot somewhere in between, and to try to find out where that sweet spot is and to regulate gene expression, because this sweet spot is moving, you regulate the gene expression so you're always tracking that sweet spot. This, I think, is one huge opportunity for cybergenetics in industrial biotechnology. Very often, the impediment is that you have intermediates that are toxic. And so by knowing what the levels of these intermediates are and adjusting variables like gene expression, you can keep the toxicity level to the bare minimum and in the process enhance the yield. So we have in my lab bioreactors or photobioreactors. So these are bioreactors that we have outfitted with LEDs, and we shine the light in feedback to control the entire population. We're not doing single cell control, but we are looking at whole population level control. The other application I'm really excited about for synthetic biology is the therapeutic applications, so the medical implications. And that's more long-term and more challenging. It takes longer time and also has lots of challenges. I mean, you want to have, anytime you put in a modified cell in a human, you need to get FDA approval. It's a complex and long process. But it's also extremely promising. So one example of this is, I think 2017 or 18, FDA approved the use of CAR T cells for immunotherapy. And this has been one of the most successful treatments for cancer, right? So immunotherapy. So these are cells that are genetically modified to attack cancer cells. And essentially, it allows immune response, immune system to target cancer cells. And these cells originate from the patient or from a donor. They're modified in the lab and then put back into the patient's blood so that they can treat diseases. So the problem is that all of these systems, the cell therapies, generally work in an open loop. And so the idea is to actually bring in some of these controllers that we've been developing and have these cells work in a closed loop fashion so that they're sensing the disease state and then producing the amount of therapy or therapeutic, just the right amount to regulate the level of, let's say, a particular biomarker, disease biomarker. And so that's the goal that we have in the lab, and that's what we're working towards. We're making better and better PID controllers or PI controllers, and then adding safety features to them so that they can become, let's say, medical grade, so that they have anti-wind-up schemes and, the usual things one has to worry about when implementing integrators in the feedback loop. But really, if you really think about it, most of diseases are essentially due to or can be attributed to feedback loops that have gone awry, right? So, think about cancer, think about diabetes, and so on. Many of the diseases we encounter are essentially due to the
genetic circuits going astray.
Yeah, genetic circuits for various reasons, right? And so by having the technology to replace these with genetically engineered cells that function as robust control systems, I think we would be able to contribute to disease. At least that's the guiding principle for us and for what we do. And, we will see what happens. This is a long process. It's not something that you can do in the lab today and test it on people the next day. There has to be some different trials, clinical trials, different phases, and it takes years and years. But we have to start somewhere.
This is so cool. It is science fiction, and I think it's fantastic to watch, from an external perspective. I kind of envision these things as almost like we are building genetic robots, which is incredible. And I guess it also comes with very important ethical challenges. Is this something that you thought about?
Yes, I think ethics comes with synthetic biology from day one, right? Anytime you want to modify something genetically, I think one has to think about the consequences. What we do in the lab, these bacteria, they don't survive outside the lab. So whatever modifications we have will have no consequences. However, these days, the tools are available to do much more powerful genetic changes. For example, CRISPR-Cas. I think that this is a new technology that led to a Nobel Prize that allows scientists to modify DNA with precision and efficiency. And this led to a lot of discussions about what one should be able to change or not to change. I mean, the danger, of course, is that you can have these CRISPR babies, as they're called. And there has been already some gross violations by one of the scientists who modified human cells to have, two new babies that have modified DNA. And I think that's very dangerous grounds. There's a fundamental difference between changing, let's say, regular cells and changing, for example, skin cells or liver cells or cancer cells in one patient, and changing cells that propagate, like, for example, sperm cells or egg cells, because the genetic modifications are not restricted to the individual, but they are passed on to their progeny. And that's much, much more dangerous. And I think this is something that we are not ready right now or scientists are not ready to go down that path. There are certain other diseases where actually you could do a lot of good by modifying the DNA. Let's say, for example, some patients that have genes that have various mutations that lead to diseases. And so if you can imagine modifying these genes through gene therapy, for example, and reversing or correcting these mutations to restore normal functions, I think that's a very exciting area of gene therapy. But whatever modifications are made are not passed on to the progeny. So I think this is a safer way of doing genetic engineering. There are clearly dangers, right? So one has to watch out not to make modifications to a cell or let's say, a bacterium that then becomes more lethal and escapes the lab and so on. And so we have to put in place various measures to ensure that this never happens. And I think ethics goes hand in hand with this business, right? So synthetic biology, like many other technologies, I think one has to be careful to put in place the right measures to make sure that the chances of accidents like that are basically zero. So with a lot of these, let's say, cell therapies, one hears about what's so-called kill switches. So if you put in the wrong therapy or something happens to the patient that has these genetically engineered cells, there would be the opportunity to kill the cell. So this would be additional circuitry added on top of the genetic circuit to stop everything. And so these are some of the measures people look at for trying to mitigate some of these risks. But at the same time, I mean, if you have a patient that is dying and they have exhausted all other possibilities and there's a new cell therapy available that could give them the chance to live longer or overcome the disease, I think then one has to take that into account as well in making these calculations.
Yeah, I guess it's very much context dependent. One thing I wanted to talk about, and then maybe we can move on towards the end of this episode, was a paper that I read from you recently. It's about the internal model principle. I know it has little to do with the applications, but it's something that is very close to my heart, something that I really, really like from classical control theory. And I thought it would be nice to touch on that as well before closing.
Yeah, thanks for bringing this up. This is something I've been very excited about recently. So this is a paper, I guess the PNAS paper that you're referring to appeared 2-3 months ago. So what this work addresses, and this is, again, I was talking about having to develop a new control theory that's particularly suited for biology. And this is a perfect example of that. I guess as control people, we are taught early on at the graduate level what the internal model principle of Francis and Wanham and the necessity of certain type of structures, integral feedback, and so on for tracking and rejecting various types of disturbances. These systems, though, that were addressed by this work are mostly linear systems, and they are not positive variables. They're not constrained by chemical reaction constraints. And so in biology, the game is a little bit different. Now, so for this particular work, we started by defining something called robust perfect adaptation. This was available for the literature. We were not the first to introduce it, but it's a useful notion. Essentially, it means tracking and disturbance rejection at the steady state that is robust, right? So robust to what? Robust to parameter changes, robust to topological changes in the network. And we know, for example, in the case of constant in time disturbances and signals, that integral feedback is sufficient for such a robustness, and it's necessary for linear systems. So how would one address this question if the underlying system is a bunch of molecules interacting with each other through chemical reactions? So they're positive variables. And what we were looking at is how do you define robustness in this case? And we looked at a particularly strong notion of robustness. We called it maximal robust perfect adaptation. In other words, the system has to be robust to changes in all the variables, all the parameters, all the reaction rates, except for the ones that determine the set point, like the dial that tunes in what the reference should be, right? You can't be robust to that. Shouldn't be even. So that's a maximal robust perfect adaptation. That's what it means. So what kind of biological networks allow you to do perfect tracking that is robust to these kind of perturbations? And we managed to obtain an answer. And the answer is different depending on whether this network is deterministic or it has noise. So it's a stochastic network, with the stochastic networks controllers being more stringent. So all of the controllers that work for the stochastic network will work for the deterministic, but not the other way around. There are motifs that will work for the noise-free case that will not work, will break down in the noisy case. For the noisy case, interestingly, we found that underlying all such motifs, there is the antithetic motif at the heart as the main core component. But more importantly, so we have an algebraic test, a very simple algebraic test. You give me the network, I can look at the certain matrix and look at those properties. And then from there, I can tell you whether this network has this adaptation property or not. And so that's one of the benefits. And if it does have this property, if it does adapt, I can construct the integrator, which is usually not obvious. Very often it's a combination of molecules that interact with each other in a certain way to implement this integral feedback. And so this theory allows you to actually find the integrator, which you wouldn't be able to do otherwise. So that's why I think this is kind of nice from a control theoretic point of view. It links in classical notions of internal model principles from Murray and Wenham with sort of modern day biochemical reaction network theory, and we have a nice clean answer. I mean, there's still a lot of open problems, but at least for this particular formulation that I just described to you, the answer is clean and it's complete.
Oddly enough, I found that this revival of the, let's say, in interest on the internal model principle has appeared in many different branches of control theory. I've seen papers, coming from the neuroscience aspects. Of course, many people in the motor control area are also very interested in this concept. And as a groupie, if you want, of this concept myself, I find it very exciting. I think now is really the time to close this episode. It's going to be probably very long, so apologies to the audience, but it was, I hope, very interesting. It was definitely very interesting to me. And as I mentioned also to many other guests of the podcast, I like to ask the question of advice to future students. So people that are interfacing with the idea of continuing in this field or potentially venturing into the synthetic biology or cybergenetics field. What is your best advice?
Okay, interesting. So I'm going to give probably a non-traditional advice, but it's one I actually do believe in, and one that I was inspired by a quote from Richard Hamming. I don't know if you know Richard Hamming or not, of course. If you had an electrical engineering background.
Stroke of genius.
Yeah, and he had, the Hamming filter. He had also Hamming windows and Hamming codes and so on, and he worked with Bell Labs. So Richard Hamming was interested in the creative process and how people do research, and he actually wrote several articles on this. And one thing that specifically struck me is the following quote. Let's see if I can remember it. And he says something like this. He says, most scientists spend almost all of their time working on problems that even they don't consider great, nor do they consider these problems will lead to great work. And so he says, then if you are working on unimportant problems, you're not going to end up with important work, except by the dumbest of luck. So this may seem trivial, maybe obvious, that of course, if you don't work on important problems, you don't end up doing important work. But I think considering that many people know that the problems they're not working on are not important, then I think it behooves us to give this more thought, right? So if you're a new student that are starting to look for a PhD problem, then I think it is useful to always ask yourself, even though the problem that you're working on seems interesting and the solution approach is interesting and the tools are interesting, how relevant would be the solution? If you are to solve what you're trying to solve, what would be the impact? And if it's minimal, maybe it's a chance to look for another problem that has a more impact. And I think really few people think about this. Usually the way you end up working on certain problems by chance, you talk to somebody or you hear a talk and you say, that sounds interesting. I can use this tool or that tool. And you get involved with the problem details and it's fun and exciting. But, at the end of the day, is it relevant? Is it important? And I think it's a good idea to ask oneself every now and then, am I working on important problems? There are always exciting tools and exciting approaches, no matter what field you work on. So I think simply by addressing this question head on early, I think one could, let's say, have a better chance of having an impact once a solution is obtained. So that's my advice is, ask yourself, is this an important problem? Because if it's not an important problem, it's not likely to lead to important results. I'm not saying it will for sure not lead to important results. Sometimes, you might run into something that ends up being important. You didn't intend to solve that problem, but it's just serendipity kicks in, right? Still, I think to the extent that you have an impact on what problems you choose, I think it's a good idea to think about the potential impact of a solution.
Well, Mustafa, with this, I think we can close this episode. Thank you so much for joining us on the show.
Thank you very much, Alberto. It's great to be here on your program.
Thank you for listening. I hope you liked the show today. If you enjoyed the podcast, please consider giving us 5 stars on Apple Podcasts, follow us on Spotify, support on Patreon or PayPal, and connect with us on social media platforms. See you next time.